Hello and welcome back to part 2 of our discussion on epidemiology. So I've given you this slide and put it in the handouts, but we're going to just basically look at pathogens, hosts, and environment and how those different things affect epidemiology. So first looking at the pathogen, we've studied virulence factors, and so virulence is the ability to cause a disease. And so these are going to be factors of an infectious agent that allow it to attach to the host cell. So maybe something like pili to avoid immune defenses, so avoiding phagocytosis, and then something that would allow it to do damage to the host. So we talked about all those different toxins, so exotoxins, and also we talked about endotoxin of gram negatives. The dose refers to the number of pathogens that are introduced. There's a minimum number of pathogens required to produce symptoms. We talked about infectious dose 50. You saw this slide a while ago. And so infectious dose 50 is the amount or the number of microbes necessary to make 50% of the population sick. And so we learned that for Shigella, it's very low, like 10 to 100, only need to be ingested. And for something like Salmonella, then you need to ingest like a million. So obviously something with a lower infectious dose would be communicated or transferred and go through a population more quickly. This is referring to the dose that someone receives. So if you're thinking about healthcare workers or lab workers, they have the potential to be introduced to a large dosage. So it also depends on the dosage that was received. Because if only a few cells enter, the immune system can probably eliminate that organism before the symptoms appear. Another thing that determines the spread of a disease is going to be that incubation period. So that's going to influence the extent of spread, because if something has a long incubation period, that means there's a whole lot of time that someone doesn't know they're sick, that they're out in society and also capable of spreading it. So your book gave this example of 10,000 individuals who were at this ski resort, and there's contaminated water with Salmonella enterica, the serotype typhi that causes typhoid fever. Well, it has a 10 to 14 day incubation period, so everyone was skiing, they all had a great time, and then they all flew off to their homes and then infected people there. And so it was an outbreak from one particular place, but then it spread to 6 different countries. And so that's just the longer the incubation, the more rapidly a disease can spread because people are asymptomatic. Okay, so I don't know if you've ever played this game. I have a 25-year-old son who used to play this game a lot, and he brought this up to me. But think about the factors associated with these different pathogens. We've talked about a whole bunch of them. So Pandemic is a game that you can play, and you get to pick a germ. So basically you can pick if it's bacterial, viral, protist. I believe you get to pick symptoms. I don't know if you get to pick incubation period or not, but let's design the perfect germ for a second. So if something is asymptomatic or has a long incubation period, like I said, that means someone is going to be out in the populace and they're going to be spreading that germ. You want it to have a low infectious dose because, once again, that is a higher likelihood of being spread to someone else. So that's going to have a higher R naught value, that reproductive rate that we talked about, or that reproductive number. You also, too, want it to be spread by respiratory droplets and aerosols, because think about it, you can prepare your food properly, you can choose what you eat, you can choose to have sex or not, but we all gotta breathe. Other thing, too, you would want is you don't want it to kill the host too quickly. I mean, you want the host to be around because the host can also spread the disease. You also want to make sure, if you're designing a perfect germ, that you want to have multiple non-human reservoirs, because that means, the humans have become inhabitable, you can just jump back to your animal hosts. The other thing, too, I would pick a virus, because viruses, there's very few drugs to treat viruses because they are inside of our cells. And so it was funny because I was talking to my son while I was talking about this game, and he said he's never won before because it's really hard to infect Madagascar. Madagascar is, an island off of Africa. And so while we were talking, I was talking about these things, and he went ahead and put in what he could to the game. I'm sorry I didn't get a film of it. But it actually, and you don't get to pick where your disease starts, but his disease actually started in Madagascar, so he was happy. That was the first time he had ever got Madagascar, and he won. So, just hey, if you're really bored, you can go check out that game. All right, so we talked about characteristics of the pathogen, so let's talk about characteristics of the host. So one thing that's going to matter in epidemiology is immunity. So whether people have been exposed before, either naturally or through immunization, and then two, if the populace has herd immunity. And so we talked about protects those non-immune individuals in a population. And so usually if it's higher than 90%, immunity is typically sufficient. There are some things we can never gain herd immunity to due to antigenic variation. And so antigens, remember, those are things that stimulate our immune system. So if they're going to be on the surface, like surface proteins of a microbial agent. And so we specifically talked, I know we talked about Neisseria gonorrhea that just, basically cycles through their pili and just changes its antigens that way. So it always stays one step ahead of the immune system. That's why you can get gonorrhea more than once. Two, we talked about the flu. We talked about antigenic drift. So these are just slight things. And then, of course, antigenic shift is even greater. So herd immunity for some things is never going to happen. Also, the general health status of a populace. So if you have a population that's malnourished, overcrowded, fatigue, you are going to have a different epidemiology, so different spread of that particular disease. Particularly susceptible to be the poor, crowded, anywhere there's poor sanitation. Age, we know, also has to do with it. The very young and the elderly are always going to be more susceptible to diseases. The immune system in the very young isn't quite developed yet. And then two, the immune system wanes as people age. The other thing that occurs is the elderly are less likely to update their immunizations. So tetanus always requires a booster, and so this is just showing you new cases of tetanus. Usually it's going to be in the older population. So yeah, waning immune system and then failure to keep up with immunizations. That's actually one of the things they think may be protecting the very young from COVID-19 is that little kids have recently had their vaccinations, and they believe some of those vaccinations might be giving immunity, a little more disease resistance. Other characteristics, so gender. Of course, women are more likely to develop urinary tract infections just because the urethra is shorter and microbes are more likely to ascend. Two, there are also just some behavioral differences between the genders. Also, just hormonal difference could also change the epidemiology of a disease. Currently, once again, COVID-19 is affecting males more than it is females. They don't really know the cause of that. Behavioral practices, breastfeeding would provide protective antibodies to an infant. If someone smokes, they're going to be more susceptible to respiratory infections. And then two, your book brings out if someone eats sushi or raw fish, they're more likely to acquire that fish tapeworm. So, yes, habits. I think they also mentioned, just eating meat in general, just the diseases that can be transferred by improperly prepared meat products. Also, genetic background. So there is something called natural immunity. So if there's something genetically that is slightly different about a people group, so maybe a receptor is different, which is usually the case, then you could have what's called natural immunity. And so there are, if someone is from Africa, they would have a higher likelihood of having a natural immunity to malaria. If they found that in Northern European populations, because of that receptor that's necessary for the virus to attach, that CCR5, that virus cannot attach, so some people have a natural immunity to HIV. So your genetic background also comes into play. Other things, characteristics of the environment that you're talking about. So growth factors, all those things we talked about as far as microbes. So temperature, most microbes need it to be warm. That's why usually ORs are kept very cold. microbes need water and nutrients. They usually don't like radiation, light. Some require oxygen, some don't. This is showing a way to sterilize a hospital room using ultraviolet light. So two, if someone is living in overcrowding, poor sanitation, that's just going to have easier transmission, increase that chance of fecal oral route. And then two, in hospitals, antibiotic use allows the development of resistant organisms. So all of these things come into play, and epidemiology takes all these things into account. All right, so hopefully you got, let me see if I missed anything. Yep, I think that's it. All right, so the story of Jon Snow.
These days, most people are like me. They hear the name Jon Snow and they think of Game of Thrones. Or they think of that British news anchor. But before there was Jon Snow, the news anchor, and before there was Jon Snow, the fan-beloved brother of the Night's Watch, there was Jon Snow with an H, the 19th century medical doctor from England. But Dr. Jon Snow has a few claims to fame, including developing early anesthetics and administering anesthesia for the Queen while she delivered two of her kids. But mostly he is remembered for the way he fought cholera. In the 1850s, his timely action and clever thinking stopped an outbreak, and even though he was never recognized during his lifetime, he's now considered one of the founders of epidemiology. Our story begins in mid-19th century London, which was, in a word, gross. In two words, super gross. Like many cities in the 1800s, London saw a huge increase in population, and with that came a huge increase in poop, which nobody knew quite how to deal with. Sewer systems hadn't quite spread to the entire city, so in places like the Soho district, people sloshed their waste into the streets, dumped it into overflowing cesspools, or trucked it over to the Thames River, the river that notably also served as the city's primary water supply. During this period, maybe unsurprisingly, London and the rest of Europe were also being plagued with persistent outbreaks of cholera, a highly infectious, sometimes deadly diarrheal illness. And there was considerable disagreement over why. We didn't know that bacteria and viruses were at the root of most infectious diseases, so the prevailing idea was miasma theory, which said that they were passed around by bad air. This is where Jon Snow came in. He had encountered cholera before in mining populations and had come to believe the disease was not spread by air, but by ingesting stuff contaminated with human waste. After all, the miners brought their meals to work, didn't have a bathroom down there, and probably didn't wash their hands before eating. Gross. When Snow observed the situation in London, he therefore concluded that cholera was being spread by tiny fecal particles in the water, and in 1854, he got a chance to prove it. The year before, a new cholera outbreak reached a London borough near Snow's home, and it killed more than 500 people in a matter of weeks. Based on the area, Snow was suspicious of one water pump on Broad Street, specifically after his sample of the water turned up visible white flecks of organic material. So, he obtained a list of some people who had been killed by the outbreak and began talking to their families. And ultimately, he found the common factor was where those victims had gotten their water, that pump on Broad Street. Snow then took his evidence to the local officials, who agreed to take the handle off the pump to prevent people from using it. But even then, nobody really believed Snow about why, including Britain's General Board of Health. It's possible that his sample size wasn't enough to convince them, or maybe it was the fact that he couldn't prove what in the water was causing the illness. In any case, Snow was confident enough in his findings that when other cholera outbreaks appeared, he continued trying to find the contaminated water behind them. And ultimately, that led him to a more citywide discovery. At some point, Snow realized all the districts in London affected by the outbreaks had their water piped in from one of two suppliers, one that got their water from upstream of London and one that got it from downstream. He suspected the downstream water, as you might also suspect, had a bunch of sewage in it, so cholera should have been more common in the neighborhoods that drink it. To prove this, he began rifling through hundreds of parliamentary death records, and he found that areas with downstream water had 14 times more deaths from cholera during the outbreak. Now to you, and to me, that might seem like pretty conclusive evidence that sewage equals cholera, but for various reasons the idea still didn't catch on. And that was the end of that, or at least it was Snow's last published attempt at convincing the medical community that sewage caused cholera. He did continue to investigate it privately and do other kinds of research, but unfortunately, he died prematurely only a few years after these events. The thing is, though, Snow was very right. Cholera is typically a waterborne illness spread by sewage-contaminated water. It just took a few more decades of work and proof that microbes can cause diseases for scientists to finally prove and accept that. Thankfully for all of us, though, Snow's methods didn't fade into obscurity. Even if his work was rejected in the 1850s, his outbreak management strategies are still in use today. His idea to map the origin of cases is a technique that is currently saving lives, and it was so unique that it was a foundation of modern epidemiology. Looking back, Dr. John Snow may not have had a sword, he may not have had an army at his command, but he did wield mass health information and help shape a major field of science. So even though Snow wasn't celebrated during his lifetime, there's a lot to celebrate about him today, because in the end, there's no doubting that his contributions have helped save millions of lives.
All right, so let's hear one for John Snow. He does know something. All right, so we're going to look at different types of epidemiological studies, and we're going to go through it, but this is kind of the one-stop shop for you, because I think one of your learning goals is to compare and contrast the three. So I'm going to go through them, and then we'll finish with this slide, and I'll make sure you got everything you needed. So, all right, so we talk about a descriptive study. This was basically what John Snow did. So this is going to be data collected, so time, place, individuals, so the who, the where, the when, during an outbreak. So this is as things are happening. This is sort of what's going on with the COVID-19 outbreak. And that's to identify the possible risk factors. So when you're talking about persons, so age, gender, ethnicity, occupation, personal habits, previous illness, socioeconomic background, marital status, all of these things can be clues.
Who can it be now? Who can it be now? Who can it be now? Who can it be now?
Yeah, if these are getting annoying, someone really needs to tell me. Hopefully you enjoyed that. This is getting kind of long, though. Just kind of keeping you with me. So the other thing, too, is going to be the place. So geographic location helps pinpoint sources. So John Snow, he made maps. Can give clues about potential reservoirs, vectors, boundaries that might affect transmission. To tracking the time course of a disease also. So this is just a graph. It's looking at the different years and different months. And so tracking respiratory syncytial virus, so respiratory virus, and comparing it to Campylobacter, which is that #1 cause of acute diarrhea in the United States. What you come to find is that during the summer months, that's when people are having picnics and eating outside. So you're more likely to contract Campylobacter. And then when it's cold outside, people are indoors, and so they're face-to-face talking. So that's why you often see more respiratory diseases during the winter months. Also, too, vector-borne diseases, when it's warm, that's when you get the bugs. So during a descriptive study, hey, you might have heard of epi curves. Don't know if you've ever seen one before, but that would be when an epi curve that would be part of that descriptive study. So there are two different types of curves. So a propagated epidemic, and so this is an example of that. So you have that very first case, which would be called the index case. So that's that first person that ever got sick.
Inside me makes me feel uneasy, baby. I can't live without you. Tell me, what am I supposed to do?
so that's the index case. I promise I'm done now. I swear I'm done. All right, so the index case is that first person. So notice you see the index case, and then you see this gap right here. So that would be basically the incubation period. And so then now you have all the people that it was spread to, so you have this little uptick, and then you see a gap here too, so that would be an incubation period for the next group. Well, eventually, as more and more people become exposed, you don't see those little gaps anymore. And then usually it's a bell-shaped curve. The whole flatten the curve was to kind of cut this off. And so that would be a propagated epidemic. The other type of epidemic is a common source epidemic. So that's when you see a rise in cases, and it's usually from exposure to a single source of a pathogen. So example would be that bad water, so coming from that well. And so you get exposure, you've got the incubation period. So it's minimum incubation period, average incubation period, maximum incubation period. But then you just see this rapid rise. What can happen too with a common source epidemic, so you get this rapid rise, but then if it is communicable, meaning it can be spread, then it can then more have a propagated epidemic curve. Okay, so that was a descriptive study, and keep in mind descriptive studies are occurring basically while there is an outbreak going on. An analytical study is sort of the next phase, if you will. And so it's going to determine which of the identified risk factors are going to be relevant to disease spread. And so there's 3 different studies, and your book gives you this picture down here. In each of these different types of studies, what's happening is that there are groups that became sick. You have to find a non-sick group basically of equal type to compare them to. And so that's what's happening in an analytical study. And so, basically you have to take 2 groups, think of same ages, same genders, same socioeconomic group. All those things have to be held equal, if you will. And so a case control study would go back in time and sort of look back at the well group and the sick group and try to find factors that are only in the sick group and not in the well group, just to sort of determine what may have led to the onset of disease. A cross-sectional group is when you look at the different groups basically at one point in time. And so that's why it's kind of showing you it's only sort of like a snapshot in that one point in time, and basically trying to see okay, so now we have some risk factors. Are they actually associated? How are they associated with that disease? And then a cohort study would be something that is moving forward. So you take those risk factors and you're going to follow a group with those risk factors into the future, basically to see if that disease develops. And then the last type are called experimental studies. And these are most frequently used basically to assess the effectiveness. So either if it's prevention, you can think about vaccine trials. Or if it's treatment, it could be you would take a group that had the disease, whether it's going to be antibiotic therapy or chemotherapy. And so whenever you do an experimental study, there's going to be 2 groups. So you have your study population, you're going to split them. One of them is going to serve as the control. And that control usually is going to get what's called a placebo. And so that's a mock drug. It has to look the same, it has to taste the same, but it's going to have no medicinal value. And then you're going to have your experimental group that's actually going to get what you're testing. 2, these studies are often have to be what's called double-blind studies. That's to avoid bias. So neither the researchers nor the subjects know if they're receiving the real treatment or the experimental treatment. The problem with this, if you think about it, are going to be ethical issues. Someone has to be told that they are going to be placed in a double-blind study. So think about these vaccine trials. So let's say, hey, I would like to be in this vaccine trial for SARS-CoV-2. And so I go and I am in the placebo group. And so I think I'm vaccinated, so I don't really worry about it. I don't social distance. I'm going to go visit people in the hospital, and I get sick and I die. And so that's there are some ethical issues. So if the risk of being in the placebo group is too harmful, often they're going to use animals. So that's an experimental study. So definitely know what placebo is and double-blind study. All right, so yeah, this is kind of what I want you to know. So just this part, if you can kind of understand the person, place, and time of that, what I mean by that. So definitely know the difference between propagated, common source, and then two, what each type of study is looking at and what they're trying to assess. So descriptive, analytical studies, and experimental studies. Yes, oh look, yes, I made another one of these. All right, so just infectious disease surveillance. So we sort of have these three big bodies or three governmental agencies, if you will, that are involved in infectious disease surveillance. And we've all heard of the CDC. They've gotten a whole lot of play lately, but in case you're not sure, it's the Center for Disease Control and Prevention. This is a national authority, so it provides support for labs, particularly in the United States, but also abroad. It is considered a great source of information. A big thing the CDC does is it collects data on diseases that impact public health. So they collect a whole bunch of data. They also do research. They have the website, which is just for educational purposes, also for healthcare professionals. Weekly, they're going to publish what's called the MMWR, which is the morbidity. Remember, that's to do with number of people that are sick, and the mortality, the number of people have died, weekly report for the 50 notifiable diseases. And so let's take a look. And so this is, your book gives a list for 2016, but there are particular diseases that have to be reported to the CDC, and this is the list. Each state also has public health departments. So the state has, and these states have their own authority to mandate that certain diseases have to be reported. And that makes sense because there are some endemic diseases to particular regions. So we talked about the Southwest, so coccidioidomycosis. That is not going to be something that needs to be reported, in Florida per se. Two, like certain states, so Washington State had an E. coli outbreak, so now they actually require E. coli 0157H7 to be reported. They also had a crypto outbreak, so they require that to be reported. So a state can also say, hey, we want to hear about this particular disease. The state authority is going to work with public schools. Public schools report absentee rates. Hospital laboratories have to report to public health departments as well for isolation, different pathogens. And then they're going to work with the news media that are going to alert the public to the presence of an infectious disease. And we've also probably heard a whole lot about WHO. They're the World Health Organization. They are part of the United Nations. There are 193 member states. The United States is one of them. They provide worldwide guidance in the field of health. They set the global standards for health. They cooperatively strengthen national health programs, so WHO works with CDC, and they develop and transfer appropriate health technology. They also have a global report, so global epidemic and pandemic news. So when there's a pandemic, they're called in, so WHO went to China. They are also involved in those eradication programs. So they are big ones that are responsible for eradicating smallpox. They're still working on polio. It's almost there. And then the third one is guinea worm, which is a helminth. They almost have it beat. There's only been about 30 cases in 2017. They have to have, in order for something to be eradicated, it has to be gone for 3 years, so no reported cases. And the reason guinea worm was targeted, it's a parasite, just by crudely filtering water and not drinking those water fleas. And then also this particular worm, the female exits out the skin, usually the foot. So if someone has a worm coming out of their foot, they shouldn't go, sloshing around where people collect their drinking water. And so just that education has really led to that eradication. But they're the ones that go globally and try to immunize people and treat people for different diseases. All right, so emerging infectious disease, we've kind of talked about this before, but in case you forgot, that is a novel or new disease or one that has recently increased in incidence. So, SARS-CoV-2 or MERS or Ebola or Zika virus. Like I said, it could also be a disease that has been around, so increasing incidence include malaria and tuberculosis. And so, emerging in several ways, we know that microbes can evolve. Vibrio cholera 0139 now has a capsule, which it didn't have before. We talked about how E. coli is normally very harmless, but if it's the one with the Shiga toxin, then it can kill people. Avian flu, they're so concerned about that because if it can then be transferred human to human. So the big thing is microbes can change. The other thing too, complacency in public health efforts. There's only a finite amount of money that's going to go to research and different diseases. So when one disease kind of goes away, like tuberculosis, funds get diverted, and so there's been increase in tuberculosis, particularly antibiotic-resistant tuberculosis. Also, changes in human society. So just the fact of daycares, and just think of all the diaper changing that goes on there that didn't used to occur. So you got Giardia, Shigella, things like that. Just easy transfer of pathogens, also just respiratory viruses and diseases as well. Advances in technology, your book talks about contact lenses. People didn't used to stick things in their eyes and keep them there, so more eye infections. Just birth control pills led to basically sex without worrying pregnancy, so that increased the rate of STDs. Population expansion, this is probably the biggest one that's causing that. So increased contact with reservoirs of disease. Lyme disease, an example, but I think I used that stat already, that 75% of emerging diseases are going to come from animal origins, zoonoses. Development, yeah. So dams are extending the range of schistosomiasis because it increased that habitat for snails. So remember, snails are involved in that life cycle. Mass production, our food is mass produced and then shipped out to the world. So if it is contaminated, it can infect a whole lot of people before we figure that out. And so it mentioned that one cantaloupe grower basically caused a multi-state epidemic of listeriosis. But you've heard that when lettuce gets distributed, we had the bluebell recall, just that our food just gets mass-produced and then shipped out, just widespread disease. So war and civil unrest. Disruption of infrastructure, refugee camps have horrible conditions, so that too get uptick in different diseases. And then climate change, warm temperatures favor reproduction of some arthropods, so just warm weather and mosquitoes, a lot of that. And then too, heavy flooding, they're just saying whenever you have flooding, you have an increase in sewage and can also overwhelm sewage treatment plants, so increased cases of cholera. All right, we are almost done. So nosocomial or healthcare associated infections, HAIs. So this is infections acquired while having treatment in the healthcare setting. And this could be anything, a clinic, a doctor's office, physical therapy, surgical center, whatever it is. So any healthcare setting, nursing home. It is among the top 10 causes of death in the U.S. And that's because hospitals and these healthcare environments, they're densely populated. You already have a weakened populace. They're sick. That's why they're there. A lot of antibiotic use, so increased chance of antibiotic resistance. And then, of course, invasive procedures. Hospital-acquired infections affect up to 5% of patients and types of them. So a lot of them are going to be urinary tract infections, particularly when catheters are used. Anything, because a lot of these surgical procedures, they're going to go through the skin. So anything can be introduced in the bloodstream. You can get sepsis. They're already immunocompromised. So you have through catheters, surgical site infections, so wound infections, pneumonias, from ventilator use. It's the most common cause of death from infection in critically ill patients. Gastroenteritis, we talked about Clostridium difficile, so related to antibiotic use. And then just other infections, the skin, eyes, uterus, burns, other things. I just thought we kind of talked about, so I'm just kind of connecting back to some of those microbes we talked about wound infections and things that make pus. So Staphylococcus aureus, Streptococcus pyogenes, Pseudomonas. The big thing with Pseudomonas, remember, you can't kill it. It can live off of almost anything. It is also environmental, so you're not getting rid of it. Big causes of pneumonia, Klebsiella pneumoniae, and once again, Pseudomonas. Diarrhea, the big one was Clostridium difficile. Enterococcus is another one, so Enterococcus faecalis. Urinary tract infections, Pseudomonas, once again, because it can form that biofilm and it can get stuck on medical equipment. Enterococcus and E. coli, those are normal gut bacteria, but if, the urethra or anus are not that far apart. And then two, hospitals, you also end up with those antibiotic resistant strains. So MRSA, methicillin resistant Staphylococcus aureus, and vancomycin resistant Enterococcus. So those are all just some causative agents of healthcare infections. So where do these come from? So clearly from other patients. Okay, so pathogens discharged via skin cells, respiratory droplets, other bodily secretions and excretions, and so you can get something from your fellow patients. Also, just from the environment. So like I said, those gram-negative rods thrive in sinks, ventilators, toilets, particularly Pseudomonas aeruginosa. And like I said, that's the one. It's particularly resistant to many disinfectants, requires few nutrients. So urinary tract infections and then wound infections. Remember, this one has that sort of blue-green color, and the smell of it is kind of grapey. Healthcare workers can be sick, so they show up for work sick. They can also be carriers of pathogens. Like I said, particularly Staphylococcus aureus. So some people just carry it basically in their nose, pharynx, or also mostly on their skin. And so this is really a problem if that particular worker is a carrier and they work in the surgical unit, ICU, or neonatals. Two, just visitors, the number of visitors should be limited in a hospital. The patient microbiota, you have to think, these are invasive procedures. So these are microbes that, if they are where they're supposed to be, they're not causing any problem. But if you introduce them, like I said, into the bloodstream, then you get sepsis and you can get septic shock and death. Two, the other thing, these people are already immunocompromised. So you can just get basically that microbiota in the wrong place. You can also get E. coli in the urethra. So any of those things. But those are the four reservoirs of HAI. So transmission of infectious agents in healthcare is pretty much going to be the same, either by direct transmission or indirect transmission. Direct transmission by healthcare personnel. It's really important that all healthcare personnel are going to be vigilant with hand washing when they go between rooms, disinfecting, wearing gloves. The big thing is going to be avoiding contact with patient fluids of any kind and then transferring it to other patients. There's something called standard precautions. And this is the minimum infection prevention practices that apply to all areas of patient care. So regardless of suspected or confirmed infection status of the patient. And so these are supposed to occur in any setting where healthcare is delivered. So just, wearing gloves, disposing of in sharps containers, how things are laundered, wearing masks. So all of those things are covered under the standard precautions. And they're designed to prevent spread. So indirect transmission, a lot of it's going to be through this. So through medical devices. Once again, think they are often breaching that first line barrier of defense. So catheters, IV lines, ventilators, inadequately sterilized invasive instruments, things airborne. There's a lot of things that basically create airborne or droplet nuclei. Think of nebulizers or intubating somebody. So it's very difficult to control. And like I said, hospitals are going to have that reduced air pressure to basically try to control air currents away from their patients. Two, they're not going to sweep. No sweeping's going on. They're going to mop to avoid kicking up dust. And then once again, using those HEPA filters to try to keep the air as clean as possible. Okay, so I just pulled this off from the MWR, Morbidity and Mortality Weekly Report from the CDC. And one of the things, of course, they are obviously tracking COVID-19. But this, I just kind of wanted to bring it to your attention. They were talking about healthcare professionals and just saying that people were exposed. They didn't recognize that this patient had COVID-19. So this was earlier in April. 3 tested positive after that encounter. All three of these had unprotected patient contact. So exposure while performing physical examinations or during nebulizer treatments were common amongst the healthcare professionals. And so it is really important to wear that PPE, personal protection equipment. Yes, there were shortages. I'm not throwing stones at this healthcare professional, but yeah, unprotected, prolonged patient contact because you have a higher likelihood of getting that dose that's required to cause disease. Also, certain exposures, aerosol generating procedures. Two, they were talking about that doing the test with that nasopharyngeal swab makes people sneeze and cough. So a saliva test would be much safer. But just, yeah. So these guidelines that are going to, that get mandated are to protect the patients, but also protect the healthcare professionals. All right. So preventing healthcare associated infections. HIAs pose risk to patients because it increases time in the hospital, increases the price of healthcare, and in some cases causes death. As I said, they are those standard precautions set out, and the purpose is to protect those patients, but also the healthcare professionals. Most of HIAs are preventable because it's important to detect causes, which they have, and then establish policies to prevent them, which they have. And so there's just three lines. Once again, hospitals have infection control committees. The CDC is also involved in this, particularly when it's an emerging disease. They're the ones that responded and made recommendations both for Zika virus and then, of course, also for SARS-CoV-2. And then there's also a National Healthcare Safety Network, which tracks data and assesses hospitals, or in HIAs. And so the good news is that data from over 17,000 hospitals show a decrease in almost every type of hospital-acquired infection. So, once again, just by identifying the risks and establishing policies and healthcare workers sticking to these policies, those numbers went down. Whew! All right, so we are done. I hope you have a great day. And, yeah, enjoy yourselves.